A warm welcome to everybody. I think it's going to be a great hour. Hopefully by the end of the hour we'll learn a lot more about immuno-oncology and Scancell in particular. I'd also like to sincerely thank John and the team who put a lot of effort into sponsoring this event. Calculus have been a great supporter of the company over the last 8 years, and we have a really solid and productive relationship with them. So we've got a number of speakers lined up today. First speaker I'd like to introduce is Dr. Cliff Holloway. Cliff has been on board just about a month, so not even the first 100 days we can discuss yet, but he's already making a substantial impact in the company, but I'll leave it to him to tell you about that. So Cliff, over to you.
Scancell is developing innovative immunotherapies for the treatment of cancer. So this field of immuno-oncology is a rapidly evolving field within the biopharmaceutical industry. There's probably not one major pharmaceutical company that's working in the field of oncology that isn't trying to build a significant franchise in immuno-oncology today. And so when we look at the development cycle for small biotechnology companies like Scancell, it starts with great science. And great science leads to great products, and great products lead to great data. And it's usually that great data, typically clinical data, that leads to that juncture where we start to look at the commercialization of lead assets with these large pharmaceutical companies who are looking to build out their therapy pipelines with new and innovative technologies and other therapies. And as Scancell takes its products through the clinic over the next year or two, we expect to see those types of interactions with a number of pharmaceutical partners. So Scancell has 2 platforms. From those platforms, we have 4 lead products, and we are addressing multiple cancer indications. So the 2 platform technologies we call ImmunoBody and ModiTope. And the immunotherapies that derive from those platforms essentially stimulate the immune system by presenting cancer antigens. So these are the unique molecular markers on the surface of cancer cells that discern them from normal cells. And that triggers potent T cell responses, and that's the T cells that are responsible for the attacking the cancer. So within our ImmunoBody platform, we have 2 products, SCIB1 and SCIB2, and we'll hear a lot more about those in the future presentations today. And then in our ModiTope platform, we have, again, 2 products, Modi1 and Modi2. So what differentiates these 2 technologies? So before I go into that, let's just look at the cancer immunity cycle. So in cancer, the release of these cancer antigens are taken up by antigen-presenting cells or dendritic cells, which in turn prime and activate T cells, which then migrate and infiltrate the tumor where they can recognize, target, and kill the cancer. But in order for this to be effective at eradicating the cancer, it has to be a competent immune response. And that's what Scancell's technologies do. So in the case of ImmunoBody, it's the mechanism of action of how we actually present these antigens to these dendritic cells that induce these potent CD8 killer T cells. In the case of ModiTope, it's actually the antigens themselves. So Scancell is the first company to recognize the ability of citrullinated epitopes or citrullinated peptides to induce very potent CD4 killer T cells. And we have a strong patent real estate around that. And this gives potential to be a game changer in the immunotherapy arena. But the net result is the same. So we're inducing 2 separate T cell responses, but the net result is the effective killing of cancer cells. So in our development pipeline, as I mentioned, 2 ImmunoBodies, 2 ModiTopes. ImmunoBody SCIB1 and the treatment of melanoma has finished a phase 1/2 clinical study, and we'll be hearing about the clinical data from that study from Professor Patel. And we're advancing that program into a phase 2 study in the United States, which we anticipate starting towards the end of this year. SCIB2 is for the treatment of non-small cell lung cancer, and we're advancing that program in collaboration with Cancer Research UK. And our ModiTope platform, Modi1, we expect to see enter the clinic in early half of 2019, and then Modi2 following thereafter, following additional preclinical evaluation throughout this year. And then another exciting development is this area of T cell therapies through a collaboration with a company called BioNTech, which I will talk a little bit more about. So you might say at this point, Cliff, aren't there already immunotherapies in the market that are treating some of these cancers? And that's absolutely true. But there's significant opportunities for improvement on these existing therapies. In terms of new cancer therapies to come in and replace standard of care or work in combination with existing therapies, but also to look at therapies that have improved safety profile, and that's particularly important when we're looking at combination therapies. We need to demonstrate efficacy in cancers where there are significant unmet needs. Not all patients respond to all therapies. Demonstrate durable responses so that we have that the cancer does not escape the therapy, and then looking at the delay and prevention of disease. In terms of how we might implement and take advantage of those opportunities, it's really trying to identify new targets, like we're doing with our Moditope platform, or new approaches to those known targets. Improve our understanding of the cancer immune cycle, to look at the synergies in terms of how we might implement our technologies with other targeted therapies, but other immunotherapies as well, and then focus on the hard-to-treat cancers, the significant opportunity there are a number of cancers that are just unresponsive to therapies. And this is where we see our therapies playing out in the clinical landscape. So when we look at immunobodies, SCIB1, for example, we've got fantastic clinical data to show that we can prevent recurrence of disease in patients, late-stage patients who have resected disease in melanoma. And now we're looking at advancing that product into a clinical trial where we're going to combine it with a checkpoint inhibitor, and these are the drugs that take the brakes off the immune system. But we're now looking at patients who have significant tumor burden. And that's the strategy that we'll be taking with SCIB2 as well. So we see this as a potential for prevention of recurrence of disease as a monotherapy, and using them and combined with checkpoint inhibitors to improve the overall response rate to these therapies, so that, for example, in melanoma, we only see about a 30% response rate to checkpoint inhibitors, and in lung cancer, about a 20% response rate. So as you can see, there's a significant headroom for improvement by looking at combination therapies. When we look at Moditope, in this case, because of the unique mode of action, we're now looking at cancers that are typically unresponsive to immunotherapies. They're not responsive to either checkpoint inhibitors or particularly unresponsive to most therapies. And in this case, we're trying to turn these so-called cold tumors hot. And so we can actually identify these tumors and actually target those in very resistant cancers. And through the collaboration with BioNTech, we're also looking at different modalities, how we might use this technology to attack these cancers by looking at T-cell receptor therapies. And this is where we're identifying the receptors on the T-cells to direct them towards the cancer and have that cell-killing effect. So just two recent announcements I would like to talk about is two collaborations that really give a significant endorsement and validation of Scancell's technologies. In December, we announced a collaboration partnership with Cancer Research UK around our SCIB2 product for the development in non-small cell lung cancer. And then shortly thereafter, we announced a collaboration with BioNTech in this area of T-cell receptor therapies. So I'll just talk a little bit about CRUK. As you know, this is a leading charity with a mandate to bring new and innovative therapies to cancer patients. And the collaboration that we have is that they've licensed our SCIB2 product, and they will undertake the responsibility for manufacturing and conducting a clinical trial, a phase 1/2 clinical trial in combination with a checkpoint inhibitor at their cost. So we look at this as a non-dilutive way to advance this program through the clinic, but we have an option to bring that program back into the company on the back of that clinical data. And obviously there's a financial option to do that, but it's a nominal fee. And if we do not undertake to take that option, then we enter into a revenue share agreement with CRUK if they decide to commercialize that independently. In the case of the BioNTech, this is a very different collaboration. BioNTech is Europe's largest private biotech company. They have collaborations with Genentech, Bayer, Sanofi, Eli Lilly, and just recently announced an A round of $270 million. And so they're obviously a very high-profile company. And one of the technologies they have is the development of these adoptive T-cell therapies, particularly around T-cell receptor therapies. And they have entered a research collaboration with Scancell to identify T-cell receptors that are directed towards the epitopes that we've identified for our MODY1 product. And at the end of that research collaboration, they have potential to take a commercial license to that technology and advance that forward. So we look forward to working with them and finishing that research collaboration and advancing this forward. But adoptive T-cell therapies have really started to become quite a huge amount of commercial interest in the industry over the recent months. Middle of last year, there was a $12 billion acquisition of Kite Pharma by Gilead, and then more recently we saw the acquisition of Juno by Celgene for about $9 billion. So you can see there's a huge amount of interest commercially within the big pharma companies to get access to these technologies. And through this collaboration with BioNTech, Scancell now has got a foothold in that landscape. So just like to talk a little bit about the progress that Scancell has made since our last funding round in May last year. We raised about £5 million. And there's been a significant amount of news flow in advancing our products towards the clinic and announcing some significant results in the clinical data that we have around our SCIB1 product. And taking SCIB1 now into the clinic in the United States in a phase 2 clinical study, and we're hoping to file our IND with the FDA next quarter, and that will initiate the process towards starting that study towards the end of this year. And we'll hear from Professor Patel later about some of the excellent 5-year survival data that we have around SCIB1 in this resected melanoma patient population. On the MODY-TOPE side, we've seen this collaboration with BioNTech, which I think is a great endorsement around that technology. But a really exciting development just recently is that we announced that Professor Durrant has put together an amazing team to meet one of the 8 grand challenges that have been presented by CRUK to tackle these big issues in cancer. And one of those issues is around coming up with personalized vaccines that are targeted to individual patients. And Professor Durrant has put together a team of international scientists, including representatives from Genentech, ISA, and BioNTech, to meet one of those challenges. And CRUK received over 130 applicants across those 8 challenges. And the team, which Scancell is a member of, has been shortlisted to a group of 10. And if we're successful in getting an award, and we hope to hear from that CRUK later this year, that could mean up to a £20 million grant to advance that technology, which will be centered on developing a MODY3 product for the treatment of cancer connected to these personalized vaccines. So we just recently, yesterday, also announced a collaboration with ISA Pharmaceuticals. As I said, we were hoping to hear from them today. Unfortunately, can't be here, but we will be talking about that. And this is an adjuvant technology that we use to combine with our MODY1 product to boost the immune response. And we can do that in the preclinical laboratory. We've shown that we can boost the immune response from 10 to 100-fold. And we expect that to translate to improved efficacy by combining these 2 technologies together when we enter this product into the clinic. And we're advancing our other products, MODY2, by identifying the actual 6-related epitopes that will become the integral component of that product moving forward. So significant advancement around our products, patient data, patents, and also partnerships. So when we look forward over the medium short term, when we sort of over the next year to 2 years, it's really about advancement of our programs, subject to sufficient funding within the company, taking our products into the clinic and advancing them through clinical trials and clinical readouts. And we anticipate 2 of those studies reading out within the next 2 years on that trajectory. Scancell has a highly experienced team, many of whom are here today. I encourage you to meet the team when we finish the presentations in the networking session. A wealth of experience in drug development, and I'm very privileged to be working with this team and taking our products forward. So what are the near-term value drivers for the company? First and foremost, it's about getting clinical data, taking our SCIB1 product through the clinic and our MODY1 product through the clinic, and we anticipate seeing those clinical readouts within the next 2 years. It's about extending our pipeline through our research collaborations with BioNTech, and if we're successful with the CRUK Grand Challenge, again, extending our pipeline with a new cancer vaccine. looking at technology partnerships like the one we announced with ICER Pharmaceuticals that help to de-risk our developments as we take these products forward. And then clinical partnerships with clinical network groups like CR UK, patient advocacy groups, but also key opinion leaders who are running our clinical trials. And I think this enables us to understand what is the utility of our products in a clinical setting. And then last but not least is industry partnerships, identifying other companies in the industry, big pharmaceutical companies and biotechnology companies that have significant franchises in the immuno-oncology landscape and have products that we can see what are the optimum combinations of combining our products with theirs so that we can meet these unmet needs in patients.
I've been tasked with telling you about our new technology, which is called Moditope, or modified epitopes. And one of the challenges really when you're trying to induce an immune response against cancer is cancer cells look very like self. They look very like normal cells. And the immune system has been educated not to react and not to reject self. Otherwise, you're into autoimmune disease. Okay? But actually, it's not quite that black and white, because what the immune system is actually educated to look for is stress. So stress meaning a cell that's unhappy. Now that could be because it's infected, it could be because it's damaged, or it could just be because it doesn't have enough nutrients or oxygen, okay? And one of these processes called, autophagy, if a cell gets unhappy, if it's stressed in any way, what it starts doing is it starts digesting some of its cytoplasm. It puts it into a membrane vesicle, it will activate a variety of enzymes, and it will release that energy from those proteins, okay? One of the enzymes that's actually activated is called PAD, and what that does is it converts this amino acid in a protein arginine to citrulline. Okay? Now, if nothing else has happened, if there's no inflammation at that point, then that's exactly what happens. The protein will get digested and you'll get energy. But if at the same time as you've got stress, you have some degree of inflammation, it could be infection, it could be massive cell death, then you upregulate what we call this MHC molecule, MHC2, and that will present these citrullinated epitopes. And that's a red flag for the immune system. As soon as the immune system sees these citrullinated epitopes, it responds very robustly with these really exciting cytotoxic CD4 T cells. So the only thing that Scancell did that was particularly novel was recognize that tumor cells, despite what everybody thinks about them and the fact they grow and kill people, they're actually very stressed. They never have enough nutrients, they never have enough oxygen, and they have absolutely massive levels of this autophagy. Okay? So the question was, did they present citrullinated epitopes, and could we exploit them as novel targets? Clearly, I wouldn't be talking to you here today if I hadn't been right with that. And the answer to that is also, we have just got our European patent basically granted, and that's for the use of any citrullinated peptide for the treatment of cancer. Again, underpinning that we were the first group to realise the potential in cancer. Okay. So what do we actually do? We make some of these citrullinated peptides, and it's fairly straightforward. Citrulline is just like any other amino acid. It can be chemically incorporated into a peptide, so they're very easy to synthesize and they're very stable. We would immunize. These modified peptides get taken up again by these APCs that Cliff was telling you about, and they stimulate these cytotoxic CD4 T cells. These T cells then migrate into the tumor environment. Now, I've already told you that tumor cells are undergoing this autophagy anyway. They're citrullinating these proteins. But what actually happens is the APCs within the tumor will originally trigger these CD4s to release this cytokine gamma interferon. And then that upregulates, as I said, this MHC class II molecules on the surface, and suddenly they're a target for these CD4s, and these CD4s kill them very efficiently and very effectively. So what we've done is we've screened a whole variety of peptides. We've identified a whole number of epitopes, 3 that we're particularly excited about, 2 from a protein called vimentin, which is a common cytoskeletal protein, and 1 from enolase, which is a common glycolytic enzyme massively overexpressed in cancers. Now, these particular proteins are expressed very highly in triple-negative breast cancer. So triple-negative breast cancer is a cancer that does not respond to hormone or other types of therapy, so it's a very acute disease. Ovarian cancer, which does respond to chemo, but if you fail the chemo, unfortunately, it has a very dire prognosis. And osteosarcoma, okay, which again, if you don't respond to chemo, has a poor prognosis. And what we've basically shown is a combination of those 3 peptides in a very aggressive tumor model gave us 100% survival. In other words, it rejected every single tumour in every single animal. Okay. I have never in 30 years seen anything that potent with any of these vaccines. These cytotoxic CD4 cells are really amazing. We've gone on then to find a good adjuvant that we could use in the clinic. So we wanted a good stable adjuvant that would work very well with our technology. And we opted for this ISA collaboration because their Amplicant vaccine is superb. What it allows us to do is link that directly to the peptide, and that means then the adjuvant and the peptide go into the same antigen-presenting cell. So then we've got the citrulated peptide and the inflammation, and a really good way of stimulating the T-cell response. What that's allowed us to actually do is reduce the dose between 10 and 100-fold, which now means not only is cost of goods much lower, but we're into realistic doses in patient to give realistic immune responses. So this combination has really made a big difference to us. If we actually take some of these animals that have rejected the initial tumour when we've immunised, and then we give those animals new tumour with no further immunisation, you can see they effectively reject them again. This means we've established a memory response within these animals, which will reject subsequent growth of tumours. And it's a key component of any vaccine. Not only do you want to cure the tumour, you want to prevent recurrence. You want to actually make sure these people survive. So the trial that we're planning to do, we'll be doing a dose escalation. So they get immunised on 0, 3 and 6 weeks, and then they get boosted at 12 and 24 weeks. We will do 3 doses. The first dose is a dose lower than we're seeing efficacy in mice. That's from a safety perspective, just to make sure there's no unexpected toxicity in humans. The intermediate dose will be the dose that we actually use in mice, where we are expecting to see a response. But we're also going to do a higher dose just in case in humans we need to give it a bit more of a boost. We need to make sure we've got the most effective dose. We will determine the most effective dose predominantly on good T-cell responses, but also if we actually see any tumour regression, we'll know we're at the right dose, not only to stimulate the T-cells, but actually to attack the tumours. And at that dose, whichever dose it is, we will have an expansion cohort to actually look, and if we can see just 3 tumour responses, 3 tumour regressions in that cohort of 16 patients, in this group of patients that are heavily pretreated with a poor prognosis, that would be a highly significant reason to go into a bigger randomised study, well, a bigger study. But we haven't just sat with Modi-1. Modi-1's now moving into manufacture and it will go in the clinic. But there are many citrullinated proteins. There are over 37 that have been recognised to date. Okay? We've looked at 7, and all 7 have given anti-tumour responses. So we still have another 30 to go along the way. They may or may not all give anti-tumour responses, but so far it's looking good. Okay? This next cohort of 4, one of them is the enolase peptide that we have in Modi-1. But these 3 new peptides are completely new targets, okay, against completely different situations. They've all given nice anti-tumour responses singly, and we're now moving them into combinations to see again if we can get this lovely 100% survival. The interesting thing here, and the really exciting thing about Moditope is, because they're autophagy targets, they're very commonly expressed cytoplasmic antigens, very highly expressed, that you can do with a few of them out, which is why they degrade them to give you energy, but it means they're expressed by all cancers. We're not talking about tumour-specific antigens here, they're expressed by this whole range of cancers. So there is a good chance that if it works in one, it will work in them all. Perhaps not in all cancers in all patients, but there's a huge repertoire that we can actually deal with. So that's particularly exciting. Again, Cliff kindly managed the Grand Challenge. And one of the reasons that we got, so the CRUK have decided that to have a non-toxic effective vaccine would be a huge step forward in cancer. Okay? And in order to do that, they have given this 20 million basically as a grand challenge to say who can come up with an effective cancer vaccine. Okay, it really needs to be done in this field of immuno-oncology. Okay, now because we were already working with BioNTech and indeed ISA in terms of our adjuvant and our collaborations, and BioNTech worked very closely with Genentech. We all got together and decided that we thought we could answer this grand challenge. So in essence, what BioNTech and Genentech do is they'll take your tumour, they'll sequence it, they'll identify all the mutations within your individual tumour, and if any of them will stimulate a T-cell response, they make an RNA vaccine and they basically immunise. And that's a really interesting technology. It's already proven to work in some melanoma patients. But only about 10 to 20% of patients will have enough mutations for this vaccine to work. So both BioNTech and Genentech are very excited about the potential of Moditope, because as I said to you, Moditope is not just for one patient. Indeed, it's not just for one cancer. It can work in a whole range of different cancers. But clearly, at this stage, it's very novel. We need to get in the clinic. We need to prove it works. So the grand challenge is about an older technology that's working, but only in a subset of patients, and a very exciting technology, Modi-3, from Scancell, that potentially could be a game changer in terms of this field. So we're going to develop this Modi-3 basically, and that's against the cancers we've chosen to go in the grand challenge are head and neck cancer, glioblastoma, lung and pancreatic cancer, potentially because they have one of the worst survival rates for any cancers. Okay, so we've taken that on the nose, and we're going to try and go with these really difficult cancers with these two approaches. So for Scancell's perspective, not only does it get their technology exposed to key opinion leaders, because we have over 21 of the best scientists in Europe and the US on this grand challenge, so we get huge exposure, but we also take one of our lead products, Modi-3, into the clinic in a different cohort of patients. So fingers crossed, we're successful. At the moment, we've been shortlisted, as Cliff said. 10 out of the 130 groups were shortlisted. We're the only vaccine consortium that's been actually shortlisted, but they don't necessarily fund all of the grand challenges. So we still have some work to do to convince them we're the right group, but very exciting. Again, Cliff's already mentioned BioNTech. So BioNTech, as you say, one of the largest private companies. They have currently over 150 staff, all focused on immuno-oncology. So really exciting. So basically, Professor Tuheen, who's head of BioNTech, is extremely interested in Moditope. Okay? He's really intrigued by the whole concept. But what he wanted to do initially was just identify the T-cell receptors on these CD4 T-cells that were stimulating this response, and see if he could identify them and use them for adoptive T-cell transfer. Again, I couldn't resist having a science slide in here, so you have to forgive me. I never give a talk without at least one slide. What we actually do at Scancell is we found a way of stimulating just blood from normal donors, not cancer patients. And then we have this clever dye that will actually allow us to visualize the CD4 cytotoxic T cells responding to our Moditope epitopes. Because we can visualize them, we can use what we call a flow cytometer to sort these cells, and we sort one cell per well across hundreds of different wells. Now, the clever technology that basically BioNTech have is they can expand then the DNA from that one cell. They can identify the gene encoding for the T-cell receptor. They can specifically clone that out, and then they can put it in a viral vector and use that T-cell receptor in a range of other patients' T-cells. So what you can then do is take any patient with cancer, you can put in that T-cell receptor, you can then make their T-cells into cytotoxic CD4 T-cells, and you can give them back to the patient, which will go and attack the cancer. Okay? Now, this is quite cumbersome, okay, and quite expensive. So if the vaccine works, then you don't need the T-cell therapy. But there will always be a situation in cancer where perhaps your immune system is not very good, we can't induce the vaccine-induced T-cell response, and it's easier for us to actually produce the T-cells in a lab and give them back to you. Potentially very advanced cancer, potentially very late cancer. The reason that BioNTech is excited is about the target, because citrin-related antigens, as I said, once you've got that TCR recognizing, say, fermentin, you could use that in hundreds of patients and hundreds of different cancer types. Okay? Again, what we've gone on to prove that those epitopes presented by a HLA molecule that's very widely expressed, so 70% of you in this room will express HLA-DP4, so we can use those to treat all of those cancers. That's very rare with T-cell therapy. Usually it's about 10 or 20% of patients at best your therapy is capable of. And of course, because we've seen such potent anti-tumor T-cell immunity with no toxicity, they're very excited. So, conclusion here, hopefully I've convinced you. We've got this nice collaboration with BioNTech to develop T-cell therapies, and that's another option out with our vaccine, so something that we would not have done, but in collaboration with them, we can take forward. Okay? We've got this great collaboration with ICER Pharmaceuticals, where we've got their adjuvant that we can directly link, and Modi-1 is now moving into manufacturing and will be hopefully in the clinic basically by hopefully beginning of next year or half of next year. But we're not just sitting with Modi-1. Hopefully Modi-1 will cure triple negative and ovarian, but my goal is to cure all cancer. never one without ambition. So MODI2 is already there for targeting lots of other solid cancers, and MODI3 in combination with the Grand Challenge will be into a bigger range of cancers. I am not naive enough to think we will cure all cancer. There will be a cohort of patients that respond really well to this vaccine, that have high levels of autophagy, high levels of citrullination, and I hope will make a difference in terms of cancer research.
My role today is to tell you about let me advance this first so you can see what I'm talking about. The ImmunoBody platform, which is Scancell's other platform. Professor Durrance told you about MODITOPE. The ImmunoBody platform is the other main platform that they are developing. So when you're looking at new immunotherapies, I think one important thing to ask yourself is how is this different from what everyone else is doing? And there is, as you know, as analysts in this field and professionals looking at this field, there's so much work going on at the moment. I think one of the key things that Professor Durrance and her colleagues at Nottingham have been able to do is to show that a key element of any immunotherapy has to be developing high avidity responses. So it's a high avidity T-cell responses, and that's what the ImmunoBody platform allows them to do. And I'll show you why that seems to be the case. But it's only the high, and avidity is really a measure of the interaction between the T-cell and its target. And it's only those high avidity cytotoxic T lymphocytes that are able to be selected into the memory pool and to mediate tumor eradication. And the ImmunoBody platform allows people to do that, allows you to do that with CD4 and CD8 T cells. So let me show you a little bit about what that looks like. So, like everything that Scancell's doing, it's well protected by a patent, by patents for the whole platform. The T cells, the T cell epitopes can be, you can pick any T cell epitopes that you want to that target tumors. You'll see later on that Professor Patel will show you the data on the first program, which was the SCIB1 program. I'm going to talk to you about the SCIB2 program. The ImmunoBody is delivered to the tumors or delivered to the patient as a DNA plasmid that has the instructions for producing this, what we've called here a genetic antigen antibody complex. So you have the epitopes inserted into the CDR regions of the ImmunoBody. At the moment, that's delivered through a process of electroporation. There's also work underway to look at a nanovesicle delivery of that, which will be slightly easier for the patients. And as I mentioned, there's the SCIB1 program for melanoma and the SCIB2 program for lung cancer. So SCIB1, we've completed a phase 1/2 trial, planning a phase 2, and it's the SCIB2 that's in partnership with the CRUK. But I want to tell you a little bit about why this is different from other vaccine technologies. So the way this DNA plasmid is taken up into cells, there are two different pathways. So there's a direct pathway where the plasmid is taken up by the antigen-presenting cell itself, produces the antibody, and that is processed then to present to the T cells. The indirect pathway, or the cross-presentation, is through this taking up of the DNA of the ImmunoBody by another cell, in this case a smooth muscle cell. That then produces and secretes the antibody, and then that antibody is also taken up by antigen-presenting cells. And the work that's been going on in Nottingham over all these years, and it has been some time, through deleting various parts of this pathway or modifying it, Professor Durrance has been able to show that it's only when you have this cross-presentation that you get the high avidity T-cell responses, and it's only when you get those that you get the effective anti-tumor response. So the target for ImmunoBody, for SCIB2 ImmunoBody, is the NY-ESO cancer antigen. This was originally developed by Lloyd Old, or discovered by Lloyd Old at the Ludwig Institute. It's been recognized as a very prominent and highly immunogenic antigen for cancers. There have been many trials with vaccines targeting this, but they've only induced rather weak immune responses. In theory, you should be able to use this to treat a number of cancers, not only non-small cell lung cancer, but you can see here a list of potential targets. And I'll tell you a little bit about why we've chosen non-small cell lung cancer in a minute. So as Lindy was saying from the last presentation, we're designing vaccines that cover a wide range of HLA types. So this SCIB2 should be suitable for more or less for the whole population. There's no reason to select patients on the basis of their HLA type. So we picked lung cancer for the reason that lung cancer is still relatively poorly treated despite the advances that have occurred in recent years. Lung cancer divides up into two main types, non-small cell lung and small cell lung, with non-small cell lung accounting for the majority of the cancers. It accounts for over a fifth of deaths from cancer in the UK. And so it's not only a common cancer, it's also a very lethal cancer. And recently we've seen success with checkpoint inhibitors treating this and beginning to displace chemotherapy as the main form of treatment for non-small cell lung cancer. But when you dig into the numbers and understand who's really benefiting from that, only if you take the first-line therapy as a monotherapy for Keytruda, which is the main checkpoint inhibitor, one of the main checkpoint inhibitors that's been developed for lung cancer, only 30% of patients are suitable for monotherapy by pembrolizumab. They have to have a high PD-L1 expression, so that's the counter target for PD-1. And only 45% of those patients will respond. Still a high response rate in that selected group, but 45% of 30% is only 15% of patients who are benefiting. And when you look at the second line treatment, the numbers are even smaller. And when you look at some of the benefits in second line, the benefits are quite marginal. With the other checkpoint inhibitor, nivolumab, the gain in median progression-free survival is a matter of 3 weeks. So you can see that there's a huge need to, immunotherapy clearly has a potential in the treatment of lung cancer, but somehow we're not quite recognizing that full potential yet. So it's this ability to try and induce not only responses, but durable responses in patients. That's the real potential of SCIB2. And so looking at some of the experimental data now from Professor Durrant's laboratory, you can see on this, on the left-hand side of this slide, we're increasing the frequency versus a standard peptide immunization, a much higher frequency of response. And then this second graph is showing the shift, the rightward shift, the leftward shift, showing a higher avidity for the responses that are generated, so a 100-fold higher avidity than the peptide immunization. Looking at the effect this is having on tumors themselves, looking in tumor cells in vitro, you're seeing a higher expression of PD-L1, which is the counterpart. Remember that slide we showed where you need the PD-L1 expression to respond to a PD-1 inhibitor. This gives a very strong rationale for combining with a PD-1 inhibitor. If you can increase the expression of PD-L1 in tumors, you should make them more sensitive to a PD-1 inhibitor. And then two graphs here showing survival of mice treated with SCIB2, inducing obviously an anti-tumor response in the top graph here, where you're having a significant improvement in survival for these mice. But very importantly, when you're taking these mice that survive, and then from day 50, re-challenging them with additional tumor without additional vaccine, comparing them to control animals, again in that second experiment, new control animals added here, the original animals re-challenged with fresh tumor, you can see there's still a survival advantage. So there's a memory response being induced here. And that's what you need to get durable responses to immunotherapy in the clinic. And importantly, then, if we are thinking about combining a SCIB2 or any immunobody with a PD-1 inhibitor, some experimental data to show that it could work. Obviously, this is still looking at mice, but we're seeing a 100% survival of the animals who receive SCIB2 and the anti-PD-1 antibody together, compared to either agent alone or compared to the control group. So as Cliff mentioned, this is the subject, SCIB2 is now the subject of a collaboration with Cancer Research UK. And it's important to recognise, I think, that when we think about Cancer Research UK, they are a research charity. They're not just a charity thinking about giving money to cancer and to cancer patients. They are a research charity. They formed from the premier research institutions in the UK. And I think this is very much a validation, again, of Scancell's work. These are scientists who are only taking on programs. I know from my own background working in companies in the States, we've had collaborations with Cancer Research UK. You only get in the door if you've got something they think is interesting and valid. And they've obviously been very interested in the work that Professor Durrant and her colleagues have done form this collaboration. This allows Scancell, as Cliff mentioned, to effectively conduct a program without having to put the investment into that, allowing them to free up their own money to do other programs. They're not short of ideas here, so there's not like the money's not going to be used for other reasons. It allows Cancer Research UK to do the program and then having the option to buy back when you are aware of the data, once the data is finalized. So the clinical trial that they're envisioning is, we've only talked in rough outlines at the moment with Cancer Research UK about what they've done. We've suggested some designs. They're going to obviously come back with their own ideas. It would be in about a quarter to a third of patients who are positive for NY-ESO-1. The important thing about all of these programs, particularly in lung cancer, is that the responses can be readily identified. We're not talking about looking at long-term time to event endpoints. We're looking at responses here. We're looking at tumour shrinkage, which is very easy to monitor by CT analysis in lung cancer patients. We would set clear targets for what we expect. We know what the standard agents alone will do. Their response rates are well defined, and there are clear statistics then for developing a null and a test hypothesis for what you expect to happen. So we would set a response rate as a target for that trial. It's an open label trial as well, so there's no reason why you can't see what's going on as you go along through the trial. The other important thing is the rate of immune-related adverse reactions. There are a lot of combinations going on with PD-1 inhibitors now. Several of those have already stopped because of the side effects of having, in addition to the PD-1 antibodies, which alone can cause severe side effects if when we're adding other agents in, getting a side effect burden which is really intolerable for the patients. The data that you'll see from Professor Patel shortly shows really why this is a very interesting idea for combining immunobodies with PD-1 inhibitors, because the immunobody by itself is so well tolerated. So in summary, the immunobody platform does allow this ability to, it's a very differentiated platform to generate these vaccines with these high avidity T-cell responses. And the SCIB2 program allows Scancell to move into a different tumour type, lung cancer, and doing so in combination with Cancer Research UK allows them to do that in addition to all the other work that they'll be doing in the future. The animal data, all the data up to this point, checks all the boxes. Doesn't mean that it's going to work, because we all know that things don't always translate from the lab to patients, but it's checked all of the boxes in a way that's, I think, exceeded everyone's expectations. It's now a question of trying to put that into practice.
I'm a clinician and a scientist, so half my job is seeing patients with cancer, trying to develop and test novel therapies. And I've got to say, with the immuno-oncology, with the advent of immuno-oncology, this is the most exciting time to be an oncologist, a time I'd never thought 10 years ago that we'd be seeing. And that's because we're beginning to see developments which are making really big changes in my everyday clinic. I'm going to talk a bit about that in melanoma and show you some background slides to know where we started and where we're at, and then the opportunities and the data which we've got with SCIB1. There are about 2,500 deaths from melanoma. Thankfully, most patients are cured because you see a mole, you can cut it out, and you can cure it. However, those 2,500, although you've cut out the primary and you thought you might be cured, you're not. And that's because in melanoma, what happens is the cancer cells which develop in the moles of your skin, is there a pointer in that? Yep. Start invading downwards and then can invade the blood vessels or the lymph vessels and spread. And unfortunately, if your melanoma is quite thin, 1 millimeter thick, so thankfully for those people, you've got a 94% chance of being cured. However, if your melanoma is 4 or 5 millimeters thick into your skin, then cancer's already spread, and your chance of being alive at 10 years is less than 40%. And that's because melanoma can spread either in the lymphatics, and that's called stage 3 melanoma, so it's already spread, but in a proportion of people, about a third, you can still cure it with surgery. But once it's gone through to stage 4, which is lungs, brain, liver, or any of these organs, the chance of cure, unfortunately, is really bad. And these are survival graphs which I've been working with for years, but thankfully are now beginning to change. For a thin melanoma, which is stage 1, the proportion of people alive at 15 years is still really high. If you've spread to the lymph nodes, stage 3, then only half of the patients are alive. If it spread further, then actually even at 1 year, less than half the people are alive, and very, very few long-term survivors. That has been completely changed by immuno-oncology, and that's drugs that target the immune response. And what they do is they harness the body's own immune cells to try and get it to fight the cancer. And they use the mechanisms which the immune system uses to fight bacteria and viral cells. So it's the T cells which are most important. And you heard a bit about the immune cycle. I'm just going to go through that. Sorry. So if you take T cells, like Lindy has done, and tumor cells, and grow them in the lab, you can get them to specifically recognize the cancer and to kill it. And the way that works is, I'm going to do an animation just to talk you through it. These dendritic cells, or antigen-presenting cells, take bits of tumor, migrate it to the lymph node where it shows it to your T cells, activates them. Those activated T cells then go to the tumor and recognize it and can kill it. But that doesn't happen in a lot of people because the tumors make things that stop the immune system, stop the dendritic cells working. And the immune system also has a whole load of very, very intricate breaks to stop it becoming overactive. So you want the immune system to switch on, and then switch off. Otherwise, it starts attacking normal cells. And cancers use those breaks to switch off the immune system. And I'm just going to show you one of those breaks, which is CTLA-4. So the dendritic cell in the lymph node comes, switches on your T cells, but then your T cells upregulate another molecule, a break called CTLA-4, which switches off that immune system to give you really good control, tight control. And so it then downregulates. So the immune switches on and switches off quickly. If you block that with a molecule, ipilimumab is one of those, checkpoint inhibitors they're called, then you can take the brakes off and you get loads of activation. So that's happened, and ipilimumab was used, shown to show immune responses in cancer, and the survival graphs on which it was licensed, we did a trial which showed that those people who had ipilimumab with stage 4 melanoma, a proportion of them, about 20%, were long-term survivors. So, which showed that you could target the immune response and get long-term survivors. And that was a real game changer in immune responses. It does, however, have lots and lots of side effects. These are toxic drugs, and so there's a real need to try and find less toxic drugs. Just as there's CTLA-4 is a break, PD-1's the other break. And nivolumab and pembrolizumab are two drugs that have been licensed with that. If in melanoma, if you compare it to the chemotherapy we previously used to use, where the 1 year survival was less than 50%, suddenly we started seeing responses in patients surviving a year of 70% of patients. And based on that, the two drugs have been licensed in 2014, and we've been using them in standard practice. And I just want to show you what the difference that actually seems to make. So even 5 years ago, if you took chemotherapy, your chance of surviving a year was 30% with chemotherapy. And then with ipilimumab, that became about 50%. If with the PD-1s, that's now 70%. If you take it to 2 years, you're dropping down to 10% or 25%. It's now 50%. And at 5 years, it's gone from 4% to 20%. We don't have the 5-year data for pembrolizumab yet, but it's looking like about 40 or 50%. And we think a whole proportion of these are going to be long-term survivors. So suddenly, in my clinic now, I can be talking to patients with advanced melanoma, people in their 20s, 30s, 40s, that they may well die of old age, because actually a lot of those are long-term survivors. That, to me, shows the power of the immune system. Sadly, this slide also shows all those people in whom we need to do something different. And that's where I think the SCIB1 has a real effect. And the reason I think that's important is I showed you a slide earlier on where you have where the cancer makes things which stops dendritic cells working. And with SCIB1, you can vaccinate away from the site of the tumor, switch on the immune system so that when the brakes are taken off, you can work even better. What we did, I won't talk you through how SCIB1 works, but you've heard about that. What we did was we took that vaccine and took it into patients. We did a phase 1 trial, first in man, across several main centres in the UK. Patients with advanced cancer, which had either still had their cancer in situ, which we could see, or had their tumours resected. So there were 15 patients with tumour present and 20, and they received vaccines along the way. And the three main things I think to take away from that phase 1 trial was, the first bit was it's safe to give. The toxicity profile was really good. That tells me that it's a really focused immune response. The second, which I wasn't sure we would see with just a vaccine on its own, was that we actually saw tumor regression. So we saw lumps of cancer, and that's one here, patient with lung metastases. That's about a centimetre and a half lump, which you can see on a CT scan. That bit there is down and regressed. So that was telling me that not only was it setting off an immune response, but enough to cause a cancer shrinkage. And then the bit which I think is really important is in that proportion of people who had had their cancer taken off, we knew would have had microscopic deposits elsewhere, because we know from historical data that those patients do, those types of patients, that actually the 5-year survival rate was out here. This is not just survival, these people who haven't recurred, but 14 of the 16 were all fine. And this is a graphic to show what they were like. So this is before their entry, and what you're doing is you're seeing the surgery there, then you're seeing when they had their vaccine. You can see that some of them recurred and had other treatments. Those two sadly died. But here, they're patients who'd had several lumps taken out along the way, and then have had no recurrences. And that, for a non-toxic vaccine, suddenly makes it, maybe you could use this in patients who've had their cancer taken out at the beginning, who thought they were cured, but have got a big chance of relapsing, to treat them. And those studies have been done in large studies with ipilimumab, with nivolumab, and with some other targeted therapies. These are large randomized studies, so this is you can't do a comparison scientifically on this, but it's just to give you an idea of the proportion of patients who here, with that large study of ipilimumab, 34% of patients had stage 3, if you took the stage 3 disease, what proportion they were, and then looking at their long-term survival. So it's comparable, but you can't, it's not scientifically valid to compare those. So I think there's a real opportunity to try and take this forward into the adjuvant setting. And then also, I think seeing those tumor responses, the question then is, should you be able to combine these with ipilimumab and nivolumab in particular, or pembrolizumab, the PD-1s, to get an even better response. And the data Lindy has in mice show that that combination certainly does do that. And so that's the control group. That's people who had mice who had SCIB1. That's the anti-PD-1. But if you combine the two, you get a really good response. And that's the basis of the phase 2 trial, which is going to start in the US. Basically, patients are going to have a combination to see how much better that is than on its own.